In this portion of the training video, we will review internal quality controls and procedures for performing external control sample testing with the KeyPath MRSA/MSSA blood culture test. Internal quality controls. Each KeyPath test includes 3 types of procedural controls. Color coding. KeyPath test components are color-coded to provide easy identification when mixing reagents and while dispensing the incubated reaction tube mixtures into the sample wells on the detector. Internal positive and negative procedural controls. Each test includes 2 internal positive procedural controls that result in 2 types of control lines. The alignment line A indicates that the blue ID and the red RS reagents were dispensed into their corresponding blue and red ID and RS sample wells. When correctly applied, the alignment line A develops in the blue ID read window of the test. If the alignment line A does not develop in the blue ID read window, or if it develops in the red RS read window, the test is invalid. The control line C indicates that internal components of the reagents in both the reaction tube and the detector are working properly. Control lines should appear in both the blue ID read window and the red RS read window on the detector. A clearing of the background color in both read windows provides an internal negative control. If no interfering substances are present in the test sample and the detector is working properly, the background will be light pink to white. Background color should not hinder reading of the test. Microphage recommends that you record these controls with each test performed. External controls. Positive and negative external controls ensure that the test reagents are working properly and that the test is correctly performed. Quality control requirements should be established in accordance with local, state, and federal regulations and accreditation requirements. Minimally, it is recommended that positive MRSA and MSSA controls and a negative control be run with each new product lot or shipment and with each new untrained operator. To perform the external control procedure, you will need the following materials in addition to the materials discussed earlier for performing a patient sample. Tryptocase soy agar culture plates with 5% sheep blood or TSA-2, McFarland suspension tubes, and the means to measure a 0.5 McFarland concentration sample, and a fixed or adjustable 30 microliter volume pipette. In addition, you will need control strains for MRSA, MSSA, and a negative control strain. Microphage has validated 3 commercially available ATCC control strains that can be used for this test. External QC sample preparation. The first step in performing external quality control testing is to prepare each of the 3 QC strains for testing. Begin by culturing and isolating individual colonies of each control strain on tryptocase soy agar culture plates with 5% sheep blood. Incubate these plates overnight at 35 degrees Celsius. Use a fresh culture that has incubated for 24 hours or less for testing. Next, prepare a 0.5 McFarland suspension for each control strain. From here, you will follow the same testing process as for patient specimens, with one important exception. When inoculating the reconstituted reaction tubes with these samples, you will use 30 microliters of each control suspension instead of 10 microliters as used previously in the patient sample testing procedure. Once the reaction tubes are inoculated, begin the incubation step and complete the test procedure as you would a patient specimen. Once the testing process is complete, interpret and record the results for each control test. Note that when running the external control procedure, the test lines T are sometimes lighter than you will see when testing patient samples. This is expected and is due in part to the absence of patient blood in the control samples. Also, as with patient testing, remember that relative line intensities are not relevant to the result, and that any light pink to dark red or purple line is positive. Thank you for your time today, and thank you for using the KeyPath MRSA, MSSA blood culture test.